I welcome everyone to Seminars in Oncology. We have a special session today, the Krakoff Family Lecture. So I'm Brian Wolpen from Dana-Farber, and I have the pleasure together with Andy Aguirre to welcome Rashika Pereira today for today's lecture. So I'm just going to give a brief overview of the Krakoff Family Lecture, and then Andy's going to introduce Dr. Pereira after that. So as some of you may know, this is a special lecture that we have every year. Sandra Krakoff, together with the Krakoff and Coleman families, endowed this lecture, the Krakoff Family Lecture, in memory of Anna Maria Coleman, to share advances in pancreatic cancer from others in the field, and particularly folks outside of the Dana-Farber Harvard ecosystem. And this has now been going on for more than a decade, with quite a number of illustrious speakers coming to talk with us as part of this lecture series. Today, I am sad to report that this lecture is also in honor of Sandy Krakoff, who passed away in November of 2021. As some of you may know, she was really a steadfast champion for Dana-Farber and Dana-Farber's mission, and really a treasured member of our community. She had served as a trustee at the Institute since 1995 and had really supported progress against cancer in numerous ways. In addition to being a trustee, she was a supporter of the annual fundraising events that were at Dana-Farber. She served on the executive, the science, and the gift planning committees, and was also part of the Susan F. Smith Center for Women's Cancer's Executive Steering Committee, and did a lot of work around breast cancer awareness and treatment. So we have been and continue to be very grateful to Sandy and to the Krakoff and Coleman families for all that they have done to help us advance scientific research at Dana-Farber over the years. And we are certainly honored to continue to hold this seminar in honor of Anna Maria Coleman, and especially today in memory of Sandy Krakoff. Before I turn it over to Andy, I just wanted to read a number of the people we've had come speak over the past decade or so. Really have been many of the people who've made major discoveries in pancreatic cancer, including Chris Icabuzio-Donahue, David Tuvison, Ben Stanger, Daphne Barsagi, Steve Leach, Howard Crawford, Liz Jaffe, and now today we're very fortunate to have Rashika Pereira. So again, a big thank you to the Krakoff and Coleman families, and I'll turn it over to Andy to introduce Dr. Pereira.
Well, thanks, Brian. It's my great pleasure to introduce Rashika Pereira, whom many on the call likely know. Is really fits well within that group of illustrious speakers that Brian just mentioned. It really is an outstanding scientist, teacher, mentor. She's an associate professor in the departments of pathology and anatomy at the University of California, San Francisco. She's also the chief scientific officer of the UCSF Pancreas Cancer Program, which is an outstanding pancreatic cancer research program, a number of great clinicians and researchers in the program. I had the pleasure of meeting Rashika when she was a postdoc with Nabil Bardisi, I guess over a decade ago. And even then I knew she was something special, really dynamic, enthusiastic, and just full of great ideas. She did her graduate research at Yale University, as well as the University of Melbourne in Australia. And she's won numerous awards and honors. Her career has really taken off like a rocket. She's including the Gunter Blobel Early Career Award recipient from the American Society of Cell Biology, a very prestigious DP2 award, the NIH Director's New Innovator Award, and that's been followed by several R01 awards. And she runs a very productive, extremely well-funded research laboratory at UCSF. Made multiple contributions to science, and in particular pancreatic cancer, identified the MIT and TFE transcription factors as regulators of lysosome biogenesis. More recently, also discovered that MHC class 1 is regulated by autophagy, and that you can inhibit autophagy and enhance immunogenicity of pancreatic cancer cells. Really a fundamental discovery that she made along with Alec Kimmelman, and they published really a terrific paper with important translational implications, and has continued to do great work in lysosomal biogenesis, pancreatic cancer plasticity, and a number of other areas. She's a prolific writer, has written many different papers in outstanding journals. She's also a fantastic speaker and has spoken all over the world and given really terrific lectures on lysosomal biology, pancreatic cancer, and a number of other topics. And lastly, she's really an outstanding teacher and mentor, and has a long list of trainees in her graduate students she's trained in already a very young career, but already an impressive and impactful mentor to a number of different trainees. So we're really pleased to have you here. Thanks for joining us. We're really looking forward to your talk.
Wow, thank you. Thank you so much, Andy, Brian. It's just such an honor to present this lecture and to be included in such an incredible group of past speakers. And thank you also to the Krakow and Coleman families for endowing this lecture, and of course to my colleagues for the invitation. So as Andy mentioned, I am a cell biologist at heart. I've always had an interest in processes within the cell that move proteins from one location to the other, as well as the compartments within the cell, how they communicate together to drive a very specific phenotype, whether it's in a healthy condition or in a diseased condition. And it's exciting for me to tell you about a story still unpublished, but recently accepted, which was also a collaboration with several Dana-Farber Cancer Institute members. So I'm very grateful for the collaborative spirit of this field. And only through this collaboration, I think, are we able to really make fundamental discoveries that can be translated to patients. So with that, I will start here in a comfortable place for me and several of you on this call, which is to sort of introduce pancreatic ductal adenocarcinoma to those of you in the audience that may not be familiar with this disease. I've highlighted some main areas which have been the focal points for research in PDA, and certainly my colleagues here at Dana-Farber and the Hale Family Center for Pancreatic Cancer have really contributed in critical ways and provided important insight into several, if not all, of these processes, in particular defining some of the somatic and germline mutations associated with PDA, with activating mutations in the KRAS oncogene being near universal in PDA patients, unraveling features of tumor heterogeneity and how this relates to treatment outcomes, uncovering alterations in metabolic parameters characteristic of this disease, including circulating metabolites that may be diagnostic or predictive of outcome, and then also mechanisms that mediate immune suppression that contribute to the refractory nature of PDA to agents such as checkpoint-based blockade. So ultimately, it is this diverse and complicated set of alterations that contribute to the aggressive nature of PDA, and what makes it sort of difficult also to treat it effectively. But fortunately, I think through sort of collaboration and through our collective efforts, we've really sort of helped to uncover some truly fascinating biology of PDA, with the hope of being able to translate this biology to more effective strategies to treat patients. So as I mentioned before, I've trained as a cell biologist. I've really had this interest in studying cellular trafficking pathways and how changes in organelle function can contribute to metabolic rewiring in PDA, and certainly several trafficking pathways, some of which I've highlighted, I hope you can see my cursor here, that we broadly refer to as nutrient scavenging pathways, are in fact upregulated and altered in the context of PDA, and these include autophagy, macropinocytosis, and the organelle that I will talk to you a lot about today, the lysosome. So with that, again, I will just start with just a brief overview of what autophagy is. Again, for those of you who may not be familiar, this is an evolutionarily conserved pathway all the way from geese to humans that involves the generation of this double membrane structure decorated by proteins on the membrane like LC3, which I will refer back to later on in my talk as well. It progressively grows and encapsulates all kinds of very diverse intracellular material, everything from misfolded proteins to aged or dysfunctional organelles, such as, say, mitochondria. This membrane then fuses together to form a vesicular structure called an autophagosome that then fuses with the lysosome, a degradative organelle, to ultimately digest this captured material, and certainly transporters and permeases on the surface of the lysosome then allow for exchange of these degradation products back into the cytosol to fuel biosynthetic bioenergetic programs within the cell. At the bottom here, I've just highlighted some of the steps in this pathway, as well as some of the conserved genes that are critical for building this double membrane vesicle, for docking and fusion with the lysosome, and certainly all the lysosomal proteins that are critical for degradation of this incoming material. Now, the lysosome is not just this, I've represented it here as this circle, but it's actually a far more complicated and beautiful organelle that has very important functions within the cell. Everything from being the site of pro-growth signal transduction. This is the location where the master growth regulator, mTOR complex 1, must localize to in order to be activated. It has sophisticated molecular machines that pump protons into the lumen of the lysosome in order to acidify its lumen. This is important because a lot of the enzymes, the hydrolases that are within the lumen of the lysosome only function at acidic pH. And they serve to break down every macromolecule that you can think of, from nucleic acids, sugars, lipids, and proteins. On the membrane of the lysosome are several transporters that mediate, as I mentioned before, the influx and efflux not only of cations and anions, but also the digestion products that come from breakdown of these diverse macromolecules that are delivered to the lysosome for degradation. But beyond that, there's certainly several trafficking pathways that converge on the lysosome as the final destination. And that includes autophagosomes, autophagy, as well as canonical trafficking pathways that emanate from the plasma membrane and elsewhere in the cell. And studies in cell biology have also shown how intimate the lysosome can communicate with other organelles in the cell, other metabolic organelles in the cell, such as the mitochondria, the ER, and lipid droplets through physical contacts between these different organelle structures. And defects in the lysosome, either hyperactivation or loss of function, are associated with all kinds of diseases, from cancer to aging, neurodegeneration, microbial pathogenesis and immunity, development, stem cell biology, and so on. And of course, hyperactivation of the lysosome is something that is characteristic of cancer cells and has been known for quite some time. And certainly the observations were made that, that the lysosome functions in a different way in cancer as it might do in, say, neurodegeneration and aging, where dysfunction of the lysosome is a hallmark. So with that, I'll just highlight some of the critical work that happened well before I started in this field by investigators at Dana-Farber. So Alec Kimmelman's study, when he was investigator at Dana-Farber, showed for the first time in a really seminal paper published in Genes and Development in 2011 that constitutive activation of autophagy is a hallmark of pancreatic ductal adenocarcinoma. And this occurs, it's seemingly independent of some of the upstream cues that normally would regulate whether autophagy is on or off in a normal cell. Follow-up studies from Daphne Masagi and Cosimo Comiso showed in 2013 that other nutrient scavenging pathways, such as macropinocytosis, so this is a pathway that emanates the plasma membrane, brings in extracellular proteins and lipids, again packaged into a vesicle structure, brought to the lysosome to be degraded, that this was also a scavenging pathway that was a hallmark of PDA or associated with RAS-driven cancers, and important for fueling sort of the metabolic features characteristic of PDA. And then studies that I did as a postdoc just across the way at MGH in Nabil Batizi's lab was to show that lysosomal biogenesis was upregulated in PDA. And teleologically, this would make sense if you have an increase in flux through these trafficking pathways that converge on the lysosome, it would make sense that you would perhaps need to upregulate the biogenesis of this important hub. What was unique here is that we went into further detail of how exactly do you build more lysosomes, and what are the sort of the molecular mechanisms and features that have to be in place in order to do this. So just quickly, I'll mention the transcription factors that Andy talked about in his introduction. These are the MIT TFE factors, so master regulators of lysosomal biogenesis. There are 4 members in this family that include MITF, which many of you would know, amplified oncogene in melanoma, as well as TFEB, TFE3, and TFEC. These transcription factors were described in 2009 to be master transcription factors of lysosomal biogenesis. What that means is that they are capable of recognizing a conserved promoter element within all the autophagy and lysosomal genes in order to coordinately upregulate all these genes that are necessary for building new lysosomes, as well as activating autophagy. Now, these transcription factors are regulated via post-translational modification in normal cells and are tied to the nutrient status of the cell as well as mTOR activation. And I won't go into the details of that, but it was a really elegant set of studies to show how lysosomal biogenesis, these catabolic processes, are regulated via the abundance of particular nutrients and the activation stages of these master growth regulators such as mTOR. In the context of PDA, what we were able to show is that MIT TFE factors are in fact upregulated and constitutively trafficked into the nucleus, where they bind and activate all these lysosomal genes via a PDA-specific pathway. So this explained in part how constitutive activation of autophagy could occur in the context of PDA, but also how it's tied to lysosomal biogenesis. And so these pathways function in a coordinated and more efficient way. So from here, when I started my lab almost 6.5, 7 years ago now, I wanted to start by asking sort of a fundamental question. We knew already that either genetic or pharmacological perturbation of the lysosome was sufficient to slow, in some cases, block tumor growth, and pharmacological blockade. In this case, I'm alluding to treatment with lysosomal inhibitors such as hydroxychloroquine. Again, work that was really pioneered by Alec Kimmerman's lab when he was still at Dana-Farber. And we wanted to understand, given what we know about the lysosome, what can we learn in terms of what the lysosome contributes to driving PDA pathogenesis? And perhaps even how is this broadly applicable to other cancers as well, RAS-driven cancers in particular? And we started with a sort of a simple question. Given my background as a cell biologist, I wanted to know, how different are cancer lysosomes relative to normal lysosomes? Structurally, are they different? Functionally, are they different? And then what is it that they actually digest on a regular basis? What is brought to the lysosome to be degraded in a cancer cell that may be different from a normal cell? And how might that impart some advantages to PDA cells? So to do this, we utilized a technique that takes advantage of lysosome membrane-associated proteins that can be used to immunoisolate intact lysosomes from cells. And this is a technique that was pioneered in David Sabatini's lab by these 2 papers listed here. In this case, we're using a protein such as TMM192. This is a multipass lysosomal membrane protein. Both its N and C termini point out into the cytoplasm, so we can attach fluorescent markers and affinity probes that allow us to be able to capture intact lysosomes from cells once we've ruptured them. So this is just showing you that we can express this tag. It localizes to the right place. It overlaps with known lysosomal markers such as LAMP2. And we can, by rupturing these cells in the absence of detergents, we can capture intact lysosomes on anti-HA conjugated beads. All the other organelles in the cell are lost through the flow through, and then we can elute off our lysosomes, which are then amenable to mass spec-based analysis and biochemistry-based approaches. So on the right side of this slide, I'm showing you what that looks like, what a proteomics run looks like. This is one of the first that we did in my lab many years ago now, showing you all the proteins that are enriched from isolated lysosomes from cancer cells, which is on the right side of this axis, versus all the proteins that are enriched from lysosomes isolated from a non-cancer control, whether it be HPDE, 293, or any other non-PDA cell. So on first pass, you can appreciate that there's quite a dramatic difference in the abundance of proteins that are unique to either the cancer context or the normal lysosomal context. And these proteins, we can characterize them into two simple categories. One is what we refer to as compositional proteins, so resident lysosomal proteins, enzymes, transporters, molecular machines, etc., versus cargo proteins, things that you wouldn't expect to see within the lysosome, but are brought there via these other trafficking pathways to be degraded. So these are what we call cargo proteins. So what can we, when you think of this and you see all these spots, I guess, the first thing you think about is, what is it that we can learn through this, organelle profiling effort? And what I've placed here are some of the concepts and ideas that we were initially interested in that, we've been able to publish on and derive some insight from these lysosome profiling efforts. So for example, we can start to understand what are the proteins within the cell that are targeted for elimination via the autophagy pathway. We can start to understand unique features of cancer lysosomes that impart greater health and strength that allow them to function at heightened capacity, which is a necessary feature in the context of PDA. We can start to understand how the lysosome contributes to cellular stress, whether it be stress that comes from external stresses, anti-cancer agents, and so on, which is what I'll talk to you about a little bit more today. And also to get at this question of how the lysosome contributes to metabolic rewiring, which is characteristic of PDA. So I want to, in the next couple of slides, just give you sort of a sense of what we've been able to achieve by really asking this fundamental cell biological question about what the lysosome does in the context of PDA, and how that's really sort of opened our eyes to some interesting biology, not only of the lysosome itself, but also of PDA, and how this can be translated to strategies to really come up with new ways to treat patients in the clinic. Some of those efforts also being done at Dana-Farber. So just quickly, one of the first projects that we initiated in my lab stemmed from our observation that MHC class 1 or peptides corresponding to MHC class 1 appeared to be enriched in lysosomes isolated from PDA cells, and really sort of absent from our normal lysosomal preps. And this was really intriguing to us because, as I'm sure you can all imagine, eluding immune-mediated recognition is a hallmark of all cancer cells, and they go about doing this by a number of different mechanisms, some of which I've listed here. The one that we were obviously really interested in, based on this finding that MHC class 1 was enriched in PDA lysosomes, was this failure to either produce antigen or failure to present it effectively on the cell surface. And so this was a study that we published a couple of years ago now, which was just an extraordinarily wonderful collaboration with Alec Kimmelman's group, where we were able to show that autophagy captures MHC class 1 and traffics it to the lysosome for degradation. And this is sort of nicely presented in some ways here at the top here, where if you stain for MHC class 1, you can see in normal HPDE cells, it's predominantly on the plasma membrane, as you would expect. Whereas in PDA cell lines, you can see that it localizes to these punctate spots. And here in this inset, you can really nicely see these lysosomal rings stained with LAMP2, and this, luminal MHC class 1 in green. And this is true also, we validated this obviously in patient PDA samples, showing there's, quite a dramatic heterogeneity in where MHC class 1 was expressed, with really striking punctate distribution that appeared to, that was intracellular in patient tissue as well. Intriguingly, we went on to show that there's a mechanism via which that you can capture MHC class 1. It involves post-translational modification or ubiquitination of MHC, which is recognized by an autophagy receptor MBR1 that bridges capture of MHC to the autophagy machinery via LC3. We were able to show that either suppression of the autophagy pathway, the lysosome, or specifically MBR1 was sufficient to restore MHC total levels as well as plasma membrane localization. And this then was sufficient to drive a really potent CD8 positive T cell response. And also led to synergy with immune checkpoint blockade in a series of really elegant in vivo studies that Alex postdoc Kusuke Yamamoto did in this study. Since then, there's been almost 8 or 9 papers that have come out in the last 2 years confirming these findings that autophagy suppression is really critical for eliciting a potent anti-tumor T cell response. Not only in PDA, but also in the context of other tumor types such as lung, colorectal, and melanoma. So we're really excited about, future prospects, again, some of which are happening, at Dana-Farber and in other cancer research institutes across the country that are now starting to integrate the combination of lysosomal inhibition with immune checkpoint blockade and standard of care chemotherapy. So this was, again, going back to sort of this fundamental exploration of cell biological phenomena within these cells that can inform sort of important behavior of these cells that are, not necessarily intuitive, but come from these sorts of efforts to profile this, critical organelle in the cell. Okay. So that was an example of a cargo protein being brought to the lysosome to be degraded as a mechanism of immune evasion. But we also, as I mentioned before, there are also compositional proteins that perhaps are unique in the context of PDA. And 2 of the most enriched proteins that we found belong to the same family called the Ferlin family of proteins. And these were very unusual to us. When I first saw them, I didn't know what the Ferlins were, and a little bit of digging went a long way, as it turns out. So the Ferlins belong, they're 6 family members, 5 mammalian, 1 in C. elegans, and they are highly expressed in heart and skeletal muscle, not expressed in the normal pancreas at all. And their function is thought to be at the plasma membrane, where they facilitate repair of membrane damage. So tissues such as the heart and skeletal muscle that undergo a lot of contractile forces or mechanical stress need these proteins in order to ensure integrity of the membrane. And certainly there are human disorders in which mutations in dysferlin in particular lead to a series of muscular dystrophy-like diseases called the dysferlinopathies. So why is it that now in pancreas, where we don't express the ferlins at all, would we start to see perhaps ferlin proteins being upregulated, but also that they're lysosomally localized? And again, this paper is published, came out last year, but this is a really nice example, I think, of the extraordinary ways in which cancer cells, or extraordinary means that the cancer cells go to ensure that a critical hub within the cell is protected from damage and can function at a heightened capacity. So we did a lot of experiments to prove that the ferlins are not a cargo, but are actually localized, anchored into the membrane of the lysosome and pointing outwards into the cytoplasm. This is also represented here in this IF image. So again, in HPD cells, they really don't express these ferlins at all. But if you were to overexpress them, their default localization is the plasma membrane. Not the case in PDA. If you express the same construct in PDA cells, you see it localized to punctate structures that partially overlap with lysosomes. And we're interested in understanding what this partiality means, but I won't go into the details today. And then in a series of experiments, we were able to show that lysosomes in PDA cells are much more resistant to membrane damage. And this is really a critical feature of lysosomes in the context of cancer. And of course, if you get rid of the ferlin proteins, now they are far more susceptible to membrane damage. And this was important for us to show because in the past, lysosomal inhibition happens through broad inhibition of the lysosome. So either with hydroxychloroquine or by, say, genetic perturbation of those master transcription factors. But here, if we take away this one protein that's on the surface of the lysosome, we were able to phenocopy some of those major defects that we see with these more general lysosomal perturbing strategies. Okay, so these are two examples of a cargo protein that's targeted to the lysosome that tells us about the biology of how these cells evade immune detection, as well as a compositional protein that tells us about the biology of cancer-associated lysosomes that make them just function at a much heightened capacity. So these are some of the things that we can start to understand through these efforts to profile the lysosome. Okay, so going back to sort of our interests as a lab and where we'd like to go. Today, what I want to tell you about is, again, an unpublished story, was an incredible collaboration also with Andy's lab and Joe Mankias's lab, and is related to how the lysosome helps the cell deal with stress. And what I mean by stress is we were really intrigued by several papers that had told us that if you switch off the driver oncogenic pathway in the cell, the RAS/MAP kinase pathway, paradoxically, you start to see an increase in autophagy flux. And this was true not just in pancreatic cancer, this had been observed in several other cancer types. And then four critical papers from Frank McCormick's lab, Ron DePino's lab, Martin McMahon's lab, and Channing Durr's lab all showed an increase in autophagy flux if you were to switch off RAS, MEK, or ERK. And this might seem counterintuitive to you, given that we know that this is a RAS-driven cancer, high levels of autophagy already exist in these cells. So how do we boost that even further when we switch off the RAS/MAP kinase pathway? This is an important observation because in Martin McMahon's paper, they were able to show synergy between suppressing MEK with agents such as trametinib together with treatment with hydroxychloroquine. So this is one patient that was treated in that study, heavily pretreated, that was given dose escalating amounts of trametinib and hydroxychloroquine, and then showed a really dramatic response in terms of his circulating CA19-9 levels, as well as the regression of the primary tumor within the pancreas bed, as well as several metastases that were present in the liver. So the question that we were interested in, having seen this observation, was to try to understand what is regulating these different magnitudes of autophagy activation. So we know that in normal cells, there's a low level of autophagy flux and a low level of autophagy dependence. And what I've shown you previously, work from Alex's lab and Kimmelman's lab and several others in the field have shown that in PDA, this level of autophagy flux is elevated, and accordingly, the dependence is also increased as well. Now, from the work that's from several of these papers, as well as these four papers that I mentioned, in the context of switching off the RAS/MAP kinase pathway, autophagy flux shifts even more, or increases even more, and dependency is also increased even more, explaining this really, really striking synergistic anti-tumor activity, but with co-treatment with a MEK or ERK inhibitor together with a lysosomal inhibitor. Okay, so the questions that we were really intrigued by and interested in understanding is, how do we increase autophagy even further in response to MAP kinase pathway inhibition? And what is it that this induced autophagy is doing in the context of RAS/MAP kinase pathway inhibition that might be different or confers an advantage or a survival advantage to these cells that perhaps the baseline levels of autophagy, despite being high in PDA cells, perhaps does not do. So basically, in these two different contexts, is autophagy doing the same thing, or is it doing something a little bit different? Okay. So to get at this question, the first thing that we did, and this is, really sort of a team effort between a postdoc in my lab, Miru Ravichandran, and a technician, JJ Hu. The first thing that we did was to perform RNA-seq in response to 48 hours of trametinib treatment. And I should say everything I'm going to show you in the next slides are all with MEK inhibitor treatment, but we've repeated everything with an ERK inhibitor as well as a RAS G12C inhibitor, AMG 510. And what I'm showing you here is initially what we were really struck by was this broad upregulation of the autophagy lysosomal gene signature, which is shown here in the heat map, also in the pathway analysis. And as I mentioned before, in KP4, human pancreatic cancer cells, they have almost a 10-fold upregulation in lysosomal biogenesis relative to a normal cell. But then when you treat with a MEK inhibitor, this increases even more so, as you can see here from staining for LAMP1. So how might this happen? In addition to this upregulation of lysosomal gene signatures, what we also, so before I get there, so one of the first things that we wanted to do was to go back and look at whether there's any differences in these MIT TFE factors and how they might be functioning. As I mentioned, these are master regulators of lysosomal gene expression in PDA. So what we were able to show is even in the absence of MEK treatment, we know that MIT TFE factors are constitutively in the nucleus. In response to MEK treatment, there's an increase in the levels of TFEB. And what we were able to show is that suppression of TFEB is sufficient to block this MEK-induced upregulation in lysosomal gene expression. So this is just shown here in the hashed bar. You can see these lysosomal genes are upregulated in response to 48 hours of MEK treatment. However, if you knock down TFEB, now you can see these genes, this MEK-induced upregulation is blocked, suggesting that TFEB is necessary for this MEK-induced upregulation in lysosomal genes. Okay. In coordination with this upregulation of all the lysosomal genes, what we also noticed was a dramatic downregulation of MYC and its target genes, and which is shown here. So all these MYC target genes are being downregulated, and MYC levels are also being downregulated at the protein level and the mRNA level. And this is perhaps intuitive, and I should say, all these observations we also show in tumor-bearing mice, but also in patient-derived organoids that Andy kindly provided to us from his lab. So again, this is somewhat intuitive because it's been shown by several groups that ERK activation, this is Channing Durr, Rosalie Sears, and others, have shown that ERK activation can drive MYC transcription by phosphorylation of key transcription factors responsible for MYC transcription, but also directly via mediating protein stabilization by phosphorylation of key residues on MYC protein itself. So it was obvious, it was perhaps intuitive to us that when you suppress the MAP kinase pathway, that MYC levels would go down. But what was intriguing to us is to find the connection between this upregulation of lysosomal genes and downregulation of MYC and MYC target genes. And we were interested in this because we knew that both these transcription families belong to the same superfamily. So MYC, as many of you know, binds to a canonical E-box motif. MIT TFE factors, as I mentioned before, bind to this clear element, which is a slightly larger modified variant of an E-box. And despite the fact that they are basic helix loop helix transcription factors, the crystal structure of MIT TFE factors showed slight variation in the lucid and zippered region that make them obligate homo-heterodimerizing partners of each other within their own family and not with the MYC and broader MYC superfamily. But nevertheless, there's this overlap between the binding of to these promoter elements present in all these lysosomal genes. And then in 2019, a really beautiful study from Sandra Diazio at St. Jude showed in, I think it was in HeLa cells and iPS cells and so on, that MYC can actually occupy this clear element and suppress autophagy lysosomal gene expression. And so introduced this idea of competition for binding to this clear element between the MIT TFE factors and MYC, given that this canonical E-box element is present within this clear motif. So we wanted to ask this question, whether in the context of RAS-MAP kinase inactivation, where MYC levels are dropping, now that we've eliminated the competition, so to speak, that TFEB can now freely occupy all the clear elements present within the cell, and then maximally activate autophagy lysosomal pathway. So to test this, we first did the most simplest experiment first, which is to just knock down MYC and look at what happens to the autophagy lysosomal genes. And what you can see here is that suppression of MYC alone is sufficient to cause an upregulation in these lysosomal genes. And then, of course, combined suppression of MYC and MECI treatment is sufficient to cause, an additive effect. So this sort of told us or gave us an indication that, in fact, MYC was perhaps negatively regulating these genes, or at least getting in the way of TFEB to maximally activate these genes. So we then did, the critical experiment, which was to perform chip analysis on TFE3 and MYC. We can see significant overlap between the bound genes or bound promoters that these two transcription factors are sitting at. Of course, these were turned out to be autophagy and lysosomal pathways were enriched signatures within these genes that both MYC and TFE3 were present on. And again, I should say they're likely not present at the same time, but able to nevertheless bind independently. More importantly, when we treat with a MEC inhibitor, we can now see loss of MYC binding to these promoter elements. This is nicely shown here by just looking at all the lysosomal genes as a pathway signature, whereas TFE3 occupancy is being increased. Again, eliminating the competition and then allowing TFE3 to sort of maximally occupy these clear elements associated with lysosomal and autophagy genes. We can validate this independently. We took some of the most well-annotated clear elements for these lysosomal genes. And again, we can see by CHIP qPCR that TFE3 is binding more so to these elements in the context of MECI treatment. Again, when MYC levels are decreasing in the cell. Okay. This also, an interesting question that a reviewer asked us, actually, which we did for this paper, was more broadly, is this an indication that perhaps MYC signatures and lysosomal signatures are perhaps anti-correlated if there is this competition between the two of them? And it turned out that this seems to be the case. There's certainly a trend towards, at least in these RAS-driven cancers, a trend towards an inverse correlation or anti-correlation between high levels of lysosomal activity and MYC activity. Again, suggesting that that the two of them, there's an interplay between the two to optimally activate both anabolic pathways that MYC regulates, but also catabolic pathways that the MIT TFE factors regulate. And I should say that when MYC is downregulated in response to MEC treatment, this doesn't cause MIT TFE factors to bind to MYC target genes. We don't see that happening. It only allows MIT TFE factors to more broadly activate its own gene sets. Okay. And then this is just to show, this is beautiful work that Joe did for us, which is to do whole cell proteomics and show that we had, the chromatin immunoprecipitation, the RNA-seq to show us that the bound genes are actually going up, and then the whole cell proteomics to show that those bound genes with their transcripts increasing are also increasing at the protein level as well, telling us that when we eliminate MYC, this allows the TFE3, TFEB to be able to occupy these clear elements, activate transcription at maximal in the context of RAS-MAP kinase pathway suppression. So this explains, again, these signatures that so many people have started to see in response to RAS-MAP kinase suppression. We now have a handle of how this is happening within the cell. Okay. So this is just highlighted here in our summary that, losing MYC allows for MIT TFE factors to maximally be activated. And this brought us to the second question, which is, why is induced autophagy so important to confer protection? What is perhaps this induced autophagy doing that might be a little bit different to what it does under baseline conditions? So to do this, we utilized this technique to be able to purify lysosomes and profile its contents. In this case, we're not looking at normal versus PDA, but rather we're looking at DMSO treatment versus MECI treatment, again, 48 hours, so very acute treatment. And what we noticed was that two proteins, ferritin heavy chain and ferritin light chain, which I'm sure many of you are familiar with from the beautiful work that Joe has done, which belong to this important iron storage complex called ferritin, were enriched in lysosomes isolated from MECI treated cells. So this suggested to us perhaps that the autophagy pathway that's being activated might be degrading ferritin at a higher rate in the context of MECI treatment. Okay. And this is shown in the volcano plot, and also these are the sort of the raw values, also showing that an important autophagy receptor, NCOA4, was also enriched in the MECI treated cells, more so than in the DMSO treated. Okay. So just quickly, so again, this is beautiful work that Joe did as a postdoc with Alec that showed, really for the first time, this selective autophagy pathway called ferritinophagy, which mediates degradation, autophagy-dependent degradation of ferritin to liberate iron, bioavailable iron in the cell. And this is mediated by an adaptor-like protein called NCOA4. So I won't go into the details here, just suffice to say that this is obviously, an important mechanism by which levels of iron can be regulated in the cell. We were able to show that levels of ferritin in the lysosome are increasing under MECI treated cells. There are multiple ways in which we can assay flux. This is just one of them to show that, there's much more ferritin within lysosomes when you suppress the MAP kinase pathway in PDA cells. This also correlates with more iron in the cell, which makes sense. If you're degrading more ferritin, perhaps you would start to see more iron within the cells, and this is what we can see. So again, this is our positive control by adding exogenous iron. This is MEK inhibitor treated. These are important controls to show if we suppress degradation, now we don't see this increase in iron. If we chelate iron, we don't see this increase, and we can measure it in multiple different ways, both by live cell imaging and flow cytometry. So more ferritin degradation, more iron in the cell. And we were then intrigued in trying to understand in detail, what is it that this increased liberation of lysosome-derived iron or ferritinophagy-derived iron might be doing. And one of the important functions of cellular iron is to be incorporated into iron sulfur clusters, which are critical cofactors of approximately 200 proteins in the cell that mediate nucleotide metabolism, DNA methylation, DNA replication and repair, and mitochondrial function. In the context of mitochondrial function, there are several proteins within complex 1, 2, and 3 of the electron transport chain that are critical iron sulfur cluster proteins. And so we were very much interested in understanding whether which of these pathways are potentially critical. And again, through the proteomics studies that we did with Joe, as well as an earlier study that Joe had done, published in Cell Reports, looking at cellular responses to RAS pathway suppression, we noted that mitochondrial iron sulfur cluster proteins were the primary subgroup that were being upregulated or changing in a significant way in response to RAS MAP kinase pathway suppression, whereas these other iron sulfur cluster proteins were not. So I'm skipping over a lot of information here, but just to show you sort of the most critical data. So here's just an example of some of these iron sulfur cluster proteins associated with the electron transport chain. Several of them are being upregulated in the context of MECI treatment. And again, this told us that perhaps these iron sulfur cluster proteins and mitochondria were really critical for driving changes in mitochondrial activity. So we can show this by measuring respiration, that if you suppress MEK with a MECI treatment, we can see an increase in respiration in these cells, again, consistent with enhanced complex formation, higher order complex formation, and complex 1 and complex 3 activity. Okay, so next we asked, what might happen if is ferritinophagy necessary for this increased mitochondrial activity that we're seeing? So to do this, we kept the autophagy pathway intact, but here we knocked down NCOA4, again, critical for, from Joe's work to degrade ferritin in the cell. So here what you can see is, for example, treatment with MECI causes an upregulation in these iron sulfur cluster proteins. Suppression of NCOA4 blocks this upregulation. Of course, we can rescue this by adding exogenous iron, but we lose this regulation because now we've flooded the cell with iron. Telling us that, in fact, that this ferritinophagy-mediated degradation and liberation of iron is important to drive increased expression of these iron sulfur cluster proteins, as well as the increased respiration that we're seeing. So again, here in blue shows you the MECI-induced upregulation of respiration, and then in red is what happens when you co-treat or knock down NCOA4 together with co-treatment with MECI. Now we block this induction. Okay. So from there, we wanted to do sort of the important experiment, which is to use agents that, clinically have been used in patients. Oh, sorry, before I go there, this is also just an important experiment just to show that we can rescue proliferation by adding exogenous iron. So here, in this very simple assay, we're co-treating with MECI and knocking down NCOA4. We see this, significant anti-tumor response or anti-growth response in vitro, which we can rescue with adding exogenous iron. And this is true also in terms of if we were to measure different parameters such as induction of apoptosis. Okay, so now I wanted to get into this important experiment, which was to utilize agents that are, let's say, clinically relevant or tractable, such as lysosomal inhibitors. And we can, again, phenocopy the effects of knocking down NCOA4, again, blocking, this MECI-induced induction of iron sulfur cluster proteins. Again, this same synergistic proliferative effect, which was seen in Martin McMahon and Channing De's papers. But what we're able to show is that this is completely rescued by adding exogenous iron. And as I mentioned at the start of my talk, the lysosome does a lot of things. It regulates, divalent cations and anions. It regulates interorganelle communication. It regulates pro-growth signaling, cholesterol homeostasis, regulation of amino acid abundance, as well as regulation of iron. And it's really sort of this, what we believe to be a selective autophagy pathway to restore iron levels in the cell that is critical in this context of RAS/MAP kinase pathway suppression, given our ability to rescue this phenotype just by adding exogenous iron. Okay, so I'll sort of say, this was, sort of the summary of a lot of work and this paper, it's in press, it's coming out soon. So I'm excited. You're the first audience that I'm talking about this story where this paper has actually been accepted. So that's pretty nice. What we were able to show is both how do we induce autophagy to a higher level than what is at baseline in PDA cells? What's the mechanism behind this induced autophagy? And it's taught us a lot about how these critical transcription factors interplay with each other, but also how these critical catabolic processes are fine-tuned and maintained at the most optimal level, unless there is a stress in which it needs to be increased even more. And then this increased catabolism serves to provide a very specific metabolite in the context of RAS/MAP kinase pathway, and this is iron, to maintain mitochondrial activity. And ultimately, what this allows the cell to do is to slow down its growth by diminishing sort of anabolic processes, mediate more interorganelle communication between the lysosome and mitochondria by exchange of a critical metabolite, to then rewire its metabolism in order to adapt or activate sort of early adaptive responses that allow the cell to sustain viability in order to down the line perhaps acquire a genetic alteration that then will allow the cell to then turn on anabolic processes and start to proliferate at a higher level. Okay, now right at the end, I wanted to sort of give you again, an overview of sort of some of the work we're doing. There's a lot of work in the metabolism area that I'm not going to talk about today, but everything I've showed you so far came from sort of in vitro cultured cells and our ability to sort of understand intriguing biology from looking at lysosomes just from cultured cells. But obviously there are all kinds of caveats and limitations associated with that. And so our efforts have also been to generate model systems in which we can start to interrogate lysosomal function in the context of in vivo tumor genesis and tumor evolution, let's say. So what we've been able to do is to generate an allele where we've knocked in the same lysotag, the TMM192, into the ROSA 26 locus downstream of a stop codon, and then cross this into our KPC mouse model of pancreatic cancer, this P48 Cre, RAS G12D, and P53 flox allele. And now we can start to, and this is just showing you histology and immunofluorescence images from these mice after they've started to develop disease. And, in our KPCL model, let's say, you can nicely see staining with this HA tag, expression of this lysotag specifically and exclusively within the tumor epithelia. And now we can start to, really interrogate organelle evolution in the context of disease evolution in terms of the primary tumor, but also the metastatic lesions. How does this critical organelle contribute to tumor progression and metastasis? And these are questions that we'd love to, and we're really excited to sort of to study in this context of this in vivo mouse model. And hopefully, I'll be able to come back in a while and tell you about our efforts in this space. So with that, I want to thank again the invitation to come and speak to you today. And I of course, want to highlight the incredible efforts of the team in my lab. This is us on World Pancreatic Cancer Awareness Day last November, dressed in purple. The work I showed you today predominantly comes from Miru, a really talented postdoc in my lab, and JJ, who's actually moving to Boston because she's starting graduate school at MIT in the fall. The MHC story was a collaboration between Anthony and Alec Kimmelman's group, and then the myofilin work was done by Suprit. Of course, thank you to all our collaborators, especially those at Dana-Farber, Andy and Joe, for helping us with our studies and of course our funding. So with that, I'm really happy to take any questions. Thank you everyone for your attention.
Excellent. Thank you, Ruchika. That was truly a wonderful talk and really a beautiful tour through a number of studies that you've done to help elucidate this biology. Maybe I'll start off with one question, then Andy, you can follow. One of the questions actually in the question and answers related to what you talked about right at the end, which was a question about how you see some of these organelles change in premalignant lesions and how that may sort of promote progression to invasive disease. I don't know if you want to touch on that a little bit more.
Yeah, so I would say I don't have the results. I wish I did to tell you what the answer is, but I could tell you about our efforts to try and answer that question. So we're doing everything from looking at acute inflammation, sort of in a premalignant disease, as well as this early stage PDAC. And we're able to do this compare and contrast analysis of lysosomal composition, as well as the cargoes that have been targeted. So for example, a simple question could be, when does MHC become this enriched cargo that's being targeted for degradation? Is that an early event that's happening, or is that a late stage event? And is that informative to us to know? So these are the types of things that we really want to try and understand. We're not quite there yet. And this is actually sort of a long-term collaboration that Joe and I have to sort of do this. And so we're really excited to start to understand sort of this concept of how does organelle evolution sort of influence how the tumor evolves as well.
Great, maybe time for one more question. Thanks, Ruchika, for a terrific talk. This is a question from Jenny Gee. How does one explore lysosome autophagy therapeutically? Targeting autophagy broadly will likely be toxic.
Yeah, so this is really sort of an important concept. I will say that there's a lot that we can learn from genetically engineered mouse models in which we can suppress critical core autophagy gene. So this is, it's a little bit of a sledgehammer, but we can take away a critical autophagy gene in an adult tumor-bearing mice, and show that the primary phenotype that you see is regression of the tumor prior to any toxic effects that are happening in mostly the brain, and in terms of the mice eventually die of infectious disease or defects in the brain, but that's with a genetic suppression. There's studies that have been done where you can toggle on and off these autophagy genes or treatment with chloroquine. And again, the major phenotype is suppression of the tumor well before there's any toxic effect. So there is a window of opportunity to be able to suppress autophagy of the lysosome without having sort of the negative features of sort of a chronic suppression. And that, I think, goes back to how we design the strategies when we start to implement these concepts in patients. And things like hydroxychloroquine, the reason why we're able to use them, because these are well-tolerated agents. This is an anti-malarial that many people use, and does not have major side effects, but is nevertheless to some extent effective in blocking the lysosome. Now, there are pharmacodynamics and pharmacokinetics that the pharmacodynamics and pharmacokinetics are not optimal, so there's an opportunity to develop better agents, with the caveat that you potentially might have more toxic effects. But all to say that there's certainly all the studies that have been done so far in elegant genetic models, preclinical therapeutic studies, but also in humans in clinical trials that have already occurred, there is a window of opportunity to be able to inhibit autophagy or the lysosome effectively. So I hope that answers your question.
Great. Maybe just one last question. Dave, this is from Dave Hill. Very nice talk and beautiful work. MITF expresses a number of isoforms that have the same DNA binding region, but may interact with different cofactors. And there is some isoform switching in melanoma.
Yes.
Any isoform dependent events in pancreatic cancer?
Very good. Yes. So there are 8 isoforms, and the melanoma specific isoform is actually not expressed in PDA at all. There's, I believe it's the H isoform and another one, if I remember, if my memory serves me correctly, that are more predominant in PDA. We haven't had a chance to really look at homodimerization, isoform switching, heterodimerization with other family members in the context of PDAs yet. It's actually something that we're doing right now. And partly that's because the reagents have not been so great, and isoform specific reagents are also a little bit tricky. And the fact that unlike melanoma, we have an abundance of TFEB, TFE3, and MITF that shows some intriguing patterns of expression depending on either the cell line or the tissue or the tumor that you're looking at. So there's a level of redundancy between these family members. They can do the same thing, but there's some added benefit of having some variation, or having 2 or 1 being abundant and another being expressed as well. And perhaps that goes towards the specificity for specific gene sets within the broader lysosomal autophagy genes that might be regulated by these homo or heterodimers. So very interesting question. We'd love to sort of explore that space more.
All right, there are more questions, but I think we're going to have to ask folks to reach out directly to Dr. Pereira to get them answered, as we're a bit over time.
Oh, I'm sorry about that.
This was a wonderful talk, I think enjoyed by all. So thank you, Ruchika, for coming to talk with us today. And again, a thank you to the Krakoff and Coleman families for sponsoring this yearly lecture on pancreatic cancer. Thanks everyone for tuning in.
Thank you, everyone.